Hi everyone, I am Dr. C.S. Kulkarni, MD, and I am going to teach medical biochemistry. Before I start the topic, please like, share, and if you are now new to the channel, please subscribe it so that the next video will be intimated to you. In the last session, we ended up with metabolism and function of chylomicrons. In today's session, we will start with metabolism of VLDL. Then we have metabolism of LDL and its clinical application. Then we have lipoprotein A, and lastly we have metabolism of HDL cholesterol. Good morning everyone. Let us begin the topic. Synthesis of VLDL. They are synthesized in the liver from glycerol and fatty acids and incorporated into VLDL along with hepatic cholesterol, ApoB100, C2 and E. ApoB100 is the major lipoprotein present in VLDL when it is secreted. ApoE and SC2 are obtained from HDL in plasma. Coming to metabolism of VLDL. The half-life of VLDL in serum is only 1 to 3 hours. When they reach the peripheral tissues, ApoC2 activates LPL, that is lipoprotein lipase, which liberates fatty acids that are taken by adipose tissue and muscle. The remnant now is designated as IDL, that is intermediate density lipoprotein, and contains lower degree of triacylglycerol and higher degree of cholesterol. In the diagram, we can see the VLDL, which contains ApoE, phospholipid, unesterified cholesterol, ApoC2, cholesterol ester, triglycerides, and ApoB100. In this diagram, we can see the lipoprotein pathway. First, it is taken by intestine, then into chylomicrons, then through the blood vessel, it enters the liver, then inside the liver, it enters into the blood vessel via VLDL, then it is converted to LDL. This LDL can move to liver or to the peripheral tissues, and from the peripheral tissues, it is taken back to liver by cholesterol, HDL cholesterol. The major fraction of IDL further loses triglyceride so as to be converted to LDL, that is low density lipoprotein. The conversion of VLDL to IDL to LDL is referred to as lipoprotein cascade pathway. A fraction of IDL is taken by hepatic receptors. Function of VLDL. The VLDL carries triglycerides, that is endogenous triglycerides, from liver to the peripheral tissues for energy needs. Coming to low density lipoproteins. Low density lipoproteins transport cholesterol from liver to peripheral tissues. The only apoprotein present in LDL is ApoB100. Most of the LDL particles are derived from VLDL, but a small part is directly released from liver. The half-life of LDL in blood is about 2 days. Here in this diagram, we can see LDL, which contains cholesterol ester, ApoB100, phospholipid, unesterified cholesterol, and triglyceride. Coming to metabolism of LDL and LDL receptors. LDL is taken up by peripheral tissues by receptor-mediated endocytosis. The LDL receptors are present on cells but most abundant in hepatic cells. LDL receptors are located in specialized regions called clathrin-coated pits. In this diagram, we can see clathrin-coated pits are present on LDL with cholesterol. These clathrin-coated pits through the cytosol present to the cell. Inside the cell, the outer core of the coated vesicle is taken up by recycling, and the inner core is taken by lysosome-mediated digestion, where cholesterol is deposited. Binding of LDL to receptor is by ApoB100, and uptake of cholesterol from LDL is a highly regulated process. When ApoB100 binds to ApoB100 receptor, the receptor LDL complex is internalized by endocytosis. The endosome vesicle is thus formed fuses with lysosomes. The receptor is recycled and returns to the cell surface. The LDL particle, along with apoproteins and cholesterol ester, are hydrolyzed by lysosomal hydrolases and forming amino acids and free cholesterol. The free receptors can now return to the membrane surface to bind further LDL molecules. About approximately 70% of LDL is secreted in the liver and the rest in extrahepatic tissues. Coming to the function of LDL, about 75% of the plasma cholesterol is incorporated into the LDL particles. LDL transports cholesterol from liver to the peripheral tissues. The cholesterol thus replicated in the cell has three major fates. First, it is used for synthesis of other steroids like steroid hormones, or it may be incorporated into the membranes, or it may be esterified to MUFA by an acyl cholesterol acyltransferase for storage. The cellular content regulates the further endogenous synthesis of cholesterol by regulating HMG-CoA reductase. Coming to LDL and clinical applications. LDL concentration in blood is positive correlation with incidence of cardiovascular diseases. The fraction of cholesterol is taken by macrophages. This is not regulated pathway. Instead, increasing levels of LDL or modification of LDL by glycation or oxidation increases the fraction of cholesterol taken by the macrophages. LDL infiltrates through arterial walls and is taken up by macrophages or scavenger cells. This is the starting event of atherosclerosis to myocardial infarction. When these cells become engorged with cholesterol, the foam cells are formed that they get deposited in the subendothelial space, triggering the formation of atheromatous plaque. Procoagulant changes are induced by endothelium, resulting in higher chance of thrombosis and increased amount of coronary artery disease. Since LDL cholesterol is thus deposited in the tissues, the LDL variety is called bad cholesterol. In the diagram, we can see through the liver, the cholesterol that goes to the periphery is bad cholesterol or the LDL cholesterol. On the other hand, from the peripheral tissues, the cholesterol that enters back into the liver is good cholesterol or HDL cholesterol. Here you can see the classification between LDL and VLDL. Definition: LDL is a bad cholesterol which contains high level of cholesterol, whereas VLDL is a bad cholesterol which contains high level of triglycerides. Percentage of cholesterol in LDL is 26%, whereas in VLDL it is 10%. Percentage of triglycerides in LDL is 10%, whereas in VLDL it is 70%. Main function of LDL is it carries cholesterol throughout the body, and whereas main function of VLDL is carrying triglycerides throughout the body. Blood concentration of LDL is less than 100 mg per deciliter, whereas of the VLDL it is less than 40 mg per deciliter. Coming to lipoprotein A. Lipoprotein A or LPA should not be confused with ApoA. LPA is strongly associated with myocardial infarction and sometimes called the little rascal. LPA, when present, is attached to FOB100 by a disulfide bond. In 40% of population, there is no detectable level of LPA in serum. In 20% of population, the LPA concentration in blood is more than 30 mg per deciliter, and these persons are susceptible for heart attack at a younger age. LPA is associated with heart attacks at the age of 30 or 40 years. Indians have a higher level of LPA than Western populations. LPA has a significant homology with plasminogen, so it interferes with plasminogen activation and impairs pyrolysis. This leads to unopposed intravascular thrombosis and possible myocardial infarction. In this diagram, we can see that plasminogen activates plasmin, which binds to fibrin and thus fibrin is lysed. Whereas on the other hand, LPA, if present, has homology with plasminogen. It binds to fibrin and inhibits plasminogen activation, and thus fibrin is not lysed. Coming to LDL cholesterol is bad. LDL, especially oxidized and glycosylated LDL, creates a procoagulant surface on the endothelium, causing blood clot formation. Oxidized LDL is found higher in levels in cigarette smokers, diabetes mellitus, and insulin resistance. In this classification, there is difference between ApoA and LPA. ApoA is seen in all persons, whereas LPA is seen in some persons. In ApoA, the A is always written in capital letters, whereas in LPA, the A is written in small letters. ApoA is a constituent of HDL, whereas LPA is associated with LDL. ApoA is antiatherogenic, whereas LPA acts on procoagulant surface and is highly atherogenic. Coming to high density lipoprotein. The high density lipoprotein transport cholesterol from peripheral tissues to liver. The major apoproteins present in HDL are ApoA1 and some levels of ApoA2, ApoC and ApoE. HDL serves as a plasma reservoir for ApoC and ApoE, which can be transferred to LDL and chylomicrons and back. Here ends our session. Kindly like, share and subscribe the channel. Thank you.